Welcome back. So now we've talked about signaling along the length of neurons. We've talked about signaling along dendrites and cell bodies, which are going to be using graded potentials. And then along the length of axons, there's going to be action potentials that are going to be used to communicate along the length of axons. Keep in mind, too, that neurons can be very long cells. So, for instance, the axons that control the muscles in your foot have their cell bodies sitting in your spinal cord, and then they send an axon all the way down to your foot to innervate the muscle. And so the action potential is going to have to travel all the way down your leg along that axon to control your muscle. So that gives you a sense for how quickly these action potentials have to move down the axon to be useful. So now what we want to talk about is focus more on communication between neurons. And so that's what this session will be about. And that's going to be accomplished in large part through chemical synapses, which we've talked a little bit about already, where you're going to have the terminus of an axon, the end of an axon, that's going to synapse with some other cell. Synapse meaning that it gets very, very close to it, but they don't quite touch. And the downstream cell, or what we call the postsynaptic cell, could be another neuron, certainly. It could be a muscle cell. It could be a cell from another organ. That doesn't matter. The idea is that the two cells come very close to one another. The action potential, we already know, is going to travel down the axon and come to this terminus. And in the end of the axon, we have voltage-gated calcium channels. And so that action potential is going to cause them to open, and that's going to let calcium enter the axon terminus. And that calcium is going to act as a signal that allows vesicles that are full of neurotransmitter to fuse with the plasma membrane and dump out here into what we call the synaptic cleft. So this is where the nervous system is very similar to the endocrine system. The endocrine system is going to release hormones into the blood, and here the nervous system is going to release neurotransmitters into the synaptic cleft. However, you know that the hormones that get dumped into the blood, they're going to have to travel all the way through the body and diffuse throughout the body to get to their targets. That's going to take some time. Where here we have a very small distance that the neurotransmitter has to diffuse, and it's going to be released in really large quantities into this small space, whereas we know endocrine hormones are going to become very dilute once they're put into the circulation. So that's going to make the chemical synapse very rapid. So tons of neurotransmitter is going to get released into the synaptic cleft, and then it's going to bind specific receptors, which can often be ligand-gated ion channels, and we'll talk about some more other specific examples. That's going to cause some sort of change in the downstream cell. If the downstream cell is a neuron and those are ligand-gated channels, then we're going to cause a graded potential in this downstream postsynaptic cell. Another thing to keep in mind is that then very often this synapse is going to have some way of terminating the signal by, for instance, degrading the neurotransmitter that's in the synaptic cleft or taking it back up into the axon terminus, some way of getting rid of that neurotransmitter so that if signaling happens again, it will be felt by causing another response in the postsynaptic cell. So this again comes to the same figure that we've talked about before, where we can have excitatory or inhibitory synapses. And an excitatory synapse is going to be one that causes a depolarization of the membrane in that postsynaptic cell, and we call that an excitatory postsynaptic potential. It's just a fancy name, but it kind of makes sense. That's going to be a graded depolarization. That's what synapse 1 and 2 are causing. And that's often going to be due to an influx of positive ions. Opening sodium channels, sodium will rush in. Then we can also, we've already discussed, have an inhibitory postsynaptic potential, which is going to become when we cause a graded hyperpolarization, as was seen with synapse number 3. And that's going to be due to either influx of negative ions, or like chloride, or an efflux of positive ions like potassium. And so we've already talked about how these are going to be graded potentials and that they're going to be additive. Another thing that we need to consider is the fact that that neurons can form different types of networks. So that we can have systems that are divergent, which means that one neuron can control several downstream neurons. This would be useful if you wanted to amplify a signal or if you want to control several different cells or processes. Or you can have convergent neuronal networks, which is when several neurons give inputs to a single neuron. So this would be an example when this downstream neuron is getting inputs and it's summing these graded potentials to decide whether or not to fire an action potential. So we'll be talking about these more further in the course, but just to give you an idea of the power of these neuronal networks and of these inhibitory and excitatory synapses, we're going to talk a little bit about lateral inhibition, which is this idea of neurons inhibiting their neighbors and what that can allow us to do when we're trying to perceive what's happening, in this case when something like a pencil is touching our skin. So if you touch a pencil to your fingertip, you'll see that it makes a big depression all around the tip of the pencil. But yet when you don't look even, you can sense that it is a point that's touching your skin, not something like an eraser. And so the way that your body allows you to sense that is through lateral inhibition. So this is what's shown here, where we've got this point touching the skin. It's mostly activating this second neuron, but it's also activating the sensory touch receptor neurons 1 and 3 as well. So without lateral inhibition, we would get this yellow curve where we have neuron 2 excited the most and 1 and 3 excited less. And the way that they're going to convey how excited they are is by action potential frequency. So since 2 is being activated a lot, it's firing action potentials very rapidly, versus 1 and 3, which are firing action potentials less frequently. However, what we actually perceive after we've had this lateral inhibition happen is this second situation, where 2 is still activated, but 1 and 3 are fairly highly inhibited. And that actually changes the action potential frequency such that 2 is still firing fairly rapidly, but 1 and 3 are firing much less rapidly. And that's what allows us to really feel the point of the pencil and kind of ignore the depressed skin around it. And that's what lets us know that it's something pointy on our finger and not something more dull. The circuitry that allows us to do that is shown here at the bottom of the slide with these yellow arrows showing the inputs coming in. And then we have these divergent and convergent networks happening where the red neurons are the inhibitory neurons. And so you can see that each neuron is going to inhibit its neighbors. So this middle neuron, neuron 2, is going to inhibit both 1 and 3, And 1 is going to inhibit 2, and 3 is going to inhibit 2. But since 2 is being activated so much, it's going to inhibit 1 and 3 much more. And that's what results in this middle graph and these action potential frequency, where what has happened is the contrast between the action potential frequency of 2 and neurons 1 and 3 has increased, and that's what allows us to feel the point. So I'm not so concerned that you understand lateral inhibition and the details of it, but I just wanted to give you a concrete example of how these networks can act to accomplish something that the nervous system requires. Give you a hardcore, a real concrete example of this. We're going to finish up just talking about a couple of specific types of synapses. One, to just give you a better sense of what's actually happening with synapses, and 2, because you're going to hear about these synapses all throughout the rest of the course, because they're going to be involved in controlling the organs that we're going to study. So one type are cholinergic synapses, named because in those synapses that presynaptic cell is releasing acetylcholine as a neurotransmitter. On the postsynaptic cell, there are 2 types of receptors that can be present. One are nicotinic ligand-gated ion channels. There are nicotinic acetylcholine receptors, which are ligand-gated sodium channels. And nicotinic receptors are found on skeletal muscle and in the brain, are some examples. Or you can have acetylcholine receptors that are muscarinic, which are, instead of being ligand-gated ion channels, they're G protein coupled receptors, which will bind the acetylcholine and then start some sort of signal transduction cascade that will lead to changes in ion channels or other changes in the cells that will cause the effect. So muscarinic receptors are found on the heart, smooth muscle, glands, and also in the brain. Another type of really important synapse are the adrenergic synapses, which are ones that release catecholamines, which are neurotransmitters like norepinephrine and epinephrine. Epinephrine, which is the same thing as adrenaline, that's why they're called adrenergic synapses. Again, we can have different types of receptors on the postsynaptic cell, alpha or beta adrenergic receptors, and there's different types of those, and we'll be getting into that in different tissue and organ types, which adrenergic receptors are expressed. And again, these are G protein coupled receptors that are going to act through second messenger systems, and they're going to be present on our blood vessels, in the airways of our lungs, the heart, smooth muscle, glands, very prevalent receptors and synapse types. So we've talked about graded potentials and how those are going to be summed throughout the neuron, and how then we're going to be able to communicate using synapses between neurons, and how that's transmission of information across the synapse is going to be very quick and very efficient in its effects on the postsynaptic cell.